So if we read the problem, and I'm going to try to get all these read and think about it, and then think about the process of how you solve a problem like this. It tells you that in 1961, scientists used repeating RNA segments such as UGU, UGU, GUG, UGU, GUG to build amino acid chains. Based on amino acid chains built, they assembled the codon chart shown. So the codon chart's the ones that you guys have in your packets, right? Mm-hmm. So it's the same codon chart. They use it. It looks like they're using the rectangular one and not the circular one. So then they ask you a question. A scientist investigates the effects of single base substitution mutations in a three-base DNA sequence. Thus, this three-base DNA sequence codes for an amino acid. So there's three bases. Remember, what are bases? They're going to be either A, T, G, or C because they tell you in the problem it's DNA. Then they say the table containing some of the scientist data is shown, and they give you a table. And they say fill in the original strand boxes in the table based on the amino acid produced, and then fill the mutated strand boxes in the table based on the DNA sequence. So they give you a table, and the table says DNA sequence, right? And then they give you mRNA sequence, and then they say amino acid. So, okay, so they say, well, amino acid produces tryptophan, TRP, and they ask you, what's the mRNA sequence, right? Because they want to know the original strand boxes. Did they give you CYS?
Mm-mm.
All right, so the tryptophan. So what could tryptophan possibly be? Well, when we look at tryptophan in the TRP, find TRP in here. I don't see it. Where is it? There it is. There's only one TRP, right? Only one possible mRNA can give you TRP. That's going to be UGG. So that's UGG. Now you have to assume that there's no exons or introns involved here, right? Mm-hmm. And so if you go and you go to DNA, UGG gets translated into A, right? Because A goes with U, and then C. So your DNA original sequence was ACC in the original strand. Now, when we look at the mutated strand, what they do for B, when we look at the mutated strand, what they give you is they give you the DNA sequence TCC, and they ask you, well, what amino acid would be produced, right? Well, there you going forward. So they're just giving you, they're just reading the chart, and they're making you go backwards and then forward. That's all. The instructions aren't incredibly clear. You have to figure things out with the table, but it's not really that hard, right? So TCC, well, T would turn into what in RNA?
A.
Correct. And then C?
GG.
Correct. So what's AGG? AGG is arginine, ARG. Correct?
Mm-hmm.
It's just that simple. All right, let's go to the next problem. Let's go to the first two problems, and let's just solve them. A lot of people.
I think I kind of understood this.
All right, but I want to make sure I go over it. So we look over this and we say, again, it's the same setup, the same chart. It's that rectangular chart, and it says, which diagram shows how RNA is read to produce an amino acid chain? Well, it's read every 3 is correct. There's no, so they give you a number line, and I could see how some people might get confused, and I think you absolutely read it correctly. So good job on that. when we look at it, they give you this number line. If you have these RNA sequences 1 through 9, how would it be read? It would be read every 3. Right? It would be read 1, 2, 3, 4, 5, 6, 7, 8, 9. And you chose that correctly. Now, which of the amino acid chain would be built if the RNA segment were UGU, GUU? Well, UGU is cysteine. Then GUG is, well, hold on. I have to write it out. I can't do it on the screen. They really should. I hope they allow you to use paper. Read to me the letters. UG.
UG.
So UG what?
UG.
Okay.
UG.
So it just repeats UG all the way
It's
to
12, yeah.
So 1, 2, 3, 4, 5, 6, 7, 8, 9, 12 what? It looks longer than what you have there. Just read it. Read it to me.
UG.
Okay, so that's it. So we're going to read it every 3, correct?
Mm-hmm.
So there's 3, and there's 3, and there's 3, and there's 3. I like to draw the vertical line though, because I get confused.
Mm-hmm.
All right, so UGU is cysteine. Do you agree?
Mm-hmm.
So that's cysteine. And then GUG is glycine, GLY. Then UGU is what? Cysteine. And GUG is glycine. So I would expect to see cysteine, glycine. Did I do that right?
It's no
It's
GLY.
It's UG. Are you sure we got this original sequence correct?
Mm-hmm.
Let's do it. UGU. Is that
Yes.
correct? Okay. It's every 3. So UGU is cysteine. That's correct. GUG. Oh, I messed up. GUG is valine. It's hard to read from across the table.
Mm-hmm.
I apologize. So, all right, so it's the cysteine was right. Glycine is valine. So I misread the chart. So you got to be careful, right?
Mm-hmm.
So if it's not in your answer choice, then you know you misread it. So start again, just like we did. So that's good. So it's valine and valine. Guys, what's important here is not the right answer, but that you can do this.
Mm-hmm.
If you can't do it, you're going to run into problems because they're obviously not going to give you the same questions on the test. So cysteine, valine. All right, so you chose correctly.
Yep.
You got this, huh?
Yeah.
All right.
Okay, this kind of confused me just a little bit.
All right, so that mutation stuff. So like we got DNA, a segment of DNA is shown along with a resulting amino acid chain. 3 mutant amino acid chains are shown, each resulting in from a single base pair mutation. So that's a single base pair mutation. So we'll look at it in a minute. And okay. In the original DNA segment that would cause each of the resulting mutant amino acid chains. Place a label that identifies each of the mutations into a blank box, each next to each mutant amino acid chain. You should use each label you may use each label more than once. Place only one label in each blank box. Then to give you this rectangular chart again. All right. So this is what you're filling in here, is that correct?
Mm-hmm.
And these are blank on purpose, I believe.
Mm-hmm.
And they gave you everything else, is that right?
Yeah.
So they gave you the original DNA was TAC. TAC, then they gave you ACG, and then they gave you TCT, and they gave you TTT. That's the non-mutated original DNA. And then they said, well, the original amino acid chain would be methionine, then cysteine, then arginine, then leucine. Oops, I missed one, didn't I? AAC. AAC would be leucine, and then lysine. Okay. Now, in the first mutant, they had several mutants. They don't give you the DNA, right? They didn't tell you the mutation, but mutant 1, they give you the amino acid change. So the first mutation, the methionine MET, and the second one, there was a tryptophan. So there was a mutation here, right? Because now I have a different amino acid. That means the DNA must have changed.
Mm-hmm.
So we know that. I'm just going to mark it as this is definitely a mutation here for mutant 1. And then tryptophan, then we have GLN. This is another mutation, maybe somewhere. This is different from the original amino acid. Do you agree?
Yeah.
And then what's the next one? GLN1, isoleucine, and then glutamine. So everything changed here, right? So there was something happened where everything after it changed, and it's only a single mutation because that's what they told you in the problem. Remember, you always want to write down what you're given, right? And you were given that there was just one mutation, one nucleotide mutation. Does that make sense?
Yeah.
Well, what kind of mutation is going to make, so if I have this DNA, TAC, ACG, TCT, AAC, TTT. What kind of mutation would allow me to mutate something in here? One of these can mutate. I could change the A to a T, or C to a G, or G to a A. You know, something in there. One of these three changed the positions 4, 5, or 6. One of these was either deleted, right?
Mm-hmm.
I could have deleted it. I could have. Let's just do it. Let's see what happens, right? So right now, I have this. If I deleted, let's pick one of these three. Let's say I delete C. Well, what happens to the whole sequence?
It changes.
It changes. Everything after that changed. So if I delete it, then I have T, A, C. That stays the same. But then everything after it's going to be different. A, G, T, C, T, A, C, T, T, T. So now I have very different amino acids. But I would not have one here. There'd be nothing here. Right?
Mm-hmm.
Do you agree? Because why is there nothing there? How many letters do I have?
It's only 2.
It's only 2. That's all right. It's only 2. So I wouldn't get anything. So I would, if I had a deletion, then I would get 1, 2, 3, 4 amino acids. How many amino acids did I get with this change? I had that one. 1, 2, 3. Well, I'm sorry. It would have been 1, 2, 3, 4, 5. So I only get 4 amino acids here. I'm not going to do the translation, right? Or transcription translation. I know that every 3 bases I'm going to get an amino acid, right? Mm-hmm. The only reason I would do the transcription translation is if I was doing exons and introns, but we're going to assume we're not doing that. So 1, 2, 3, 4, 5. I have 4. I would only get 4, but I'm getting 5, so it can't be a deletion. Do you agree? If I could, what else could I do to make that? By the way, this is also known as a category of mutations known as frame shift. What that means is in a frame shift mutation, there's a deletion, there's also an insertion. So if you stick something in or delete something, what it's going to do is change everything after it. Right? So if we inserted something, instead of taking out a C, what if we said TAC A C C G T C T A C T T T? Well, I would get how many? Let's count them. I'd get 1, 2, 3, 4, 5 amino acids. There'd be an extra letter, but that's fine. 5 amino acids. So that would allow me to have 5 amino acids, and they would be different. Do you agree? So that matches. An insertion would match my problem. Insertion works. Deletion doesn't work because I would lose an amino acid, and I didn't lose one. I had 5. I have 5, so it can't be a deletion. It could be an insertion. Now, what if I substituted one? What if I just changed? Let's do that. So there's my deletion, there's my insertion. Now what if I changed one? So I'm just going to change it here at the first time I notice a difference, right? Just randomly. TAC A, instead of C, what if I put a G, right? G T C T A C T T T. Well, if I look at the results, TAC would be the same, right? So, and you can see that methionine is still methionine, so that stayed the same. That's fine, that matches. But then AGG would be different, wouldn't it? So it might lead to, this could have been a silent mutation. I'd have to look at the chart, but but let's assume it does mutate this one, okay? Did it mutate everything after it? TCT, AAC, and TTT? No. But in our problem, it mutated everything after it. So substituting one would only change that amino acid. So when you have a substitution mutation where you have put a G instead of a C, that's only going to change that amino acid because it doesn't shift the frame. It doesn't move everything after it. The frame shift happens when there's a deletion or an insertion. Does that make sense? Okay. So which of these do you think it is when you're for mutant 1? What is mutant 1? What is mutant 1? Is it an insertion, substitution, or deletion? So if I inserted one, I would get a change in only one amino acid. How many of these change? So it's not that one. So if I deleted it, I'd only have 4 amino acids, not 5. So it's not a deletion. So what is the only one left?
Insertion.
Insertion. Since they only give me 3 options, right? So that's definitely an insertion. So let's look at the next mutation. Mutant 2 says, oops, let me go. Mutant 2. And the first one, read to me the amino acids for mutant 2. MET.
TRP.
TRP.
ARG.
ARG.
LEU.
L-E-U or L-L-E-U? No. Yeah, you're right. L-E-U.
And the LYS.
L-Y-S. So there was no change. Do you agree? All right, so
So it would be this substitution.
substitution. That's right. It's likely a substitution. It's for sure it's a substitution.
Yeah.
But it's something more than a substitution. I'm going to go ahead and put this on because yes, you're right. That's the only answer that matches. So this one here was an insertion. Is that correct? And our reasoning was there were 5 because a deletion would lead to only 4 amino acids, right? Here we're going to say it's a substitution because there's no change. Well, one of the things that can happen if you substitute, it's very hard for this to happen if it's an insertion or a deletion. In fact, I would argue it's statistically impossible. To have, when you have a substitution, it could be silent. So we call it a silent mutation. It's not always a silent mutation. Sometimes when you substitute, you just change that one amino acid, okay? But sometimes when you substitute an DNA, it turns out to be the same. So let's look at the chart and look at an example. We mutated this second one before, so let's mutate it again. This is an example. What if we had ACG? And we look at our chart, and those of you at home, you have your charts in your packet. So you have ACG. What if ACG would be, ACG is 3' THR, right? Is that correct? Well, if I mutated remember that wobble position I told you about back in, I told you the third position here in the codon, which is not the codon, this is a DNA, but if I change this to mRNA, it would be a codon. ACG, and I'm reading this wrong actually, because this is the DNA, ACG is the DNA. I'd have to transcribe that into UGC, correct? UGC is cysteine, okay? So that's what it is normally. Now, if I take that ACG and mutate that and just substitute A, leave A the same, C the same, but instead of ACG, I said ACA, then my RNA would be UGU, and UGU is the same amino acid. You see? See, UGC and UGU give you the same amino acid. So that's why we call it silent, because there is a change in the DNA. There is a mutation. It's a substitution, right? We substituted A in DNA instead of G, so that's substituted. So the RNA is U instead of C. So those two things change, the DNA and the RNA. But the amino acids stay the same. So the protein won't change. Does that make sense? So we have the same protein, even though there is a mutation. So we can have the same protein, even though there's a mutation in the DNA, because of this duplication, this idea that more than one, for many of these amino acids, more than one of the sequences will give you that amino acid. So that's called redundancy. You have a backup. So that helps you when you have mutations, and you're going to have mutations because it happens randomly. They don't always cause you a change. You don't want to have grow 6 arms because you accidentally, copied the DNA wrong. So it's it's a good protection that every 1/3 of your DNA can change and not change the amino acid. So it protects us against random negative mutations. So that's called a silent mutation. But remember, you could have changed this last position to ACT, and if you would have changed it to ACT, then that would have stopped it. That would have destroyed the protein, because ACT would have gave you the RNA UGA, which is a stop.
Mm-hmm.
So a substitution can definitely mutate the protein, but it could also be silent the way it is here, okay? But only a substitution could be silent. So this is further backs up the idea that mutant 2 is a substitution. You see how complicated these questions are?
Mm-hmm.
And do I agree with what they do? I mean, it sure tests you, but I really wish they wouldn't be such, so there's no reason to go to so many, especially the format, like giving you all three and having you figure it out. Like, it's just, so here we have MET, and we have CYS, and we have SER. Now it's shorter.
Mm-hmm.
It's the problem. And here's a key. Here's an issue. In this problem, there's actually more than one right answer, because you can use. They said you can. Oh no, there's only one answer, because they tell you place only one label in each blank box. You may use each label more than once. Well, because you can use it more than once, there's more than one right answer. Because as we just said, if we would have done a substitution and we would have substituted that A to a T, then we would have gotten a mutation that would have been made the protein shorter. Does that make sense? So that could have been substitution, but it could also be a deletion. Does that make sense? So we'd have to really come up with a definitive answer. We would have to look at at what's happening here. And if you look, methionine, the first one didn't change. The second amino acid didn't change, but the third one did.
Mm-hmm.
That means you had a mutation here. That mutation in that third, in one of those nucleotides in this third set of DNA, that TCT, some mutation happened that after that place in the DNA, there was a stop. It must have stopped. It mutated, so there's something changes. Where are you guys going? Is it time to go? It's time to go, guys. So after this, it got shortened, the DNA got shortened, and it changed into a stop. So how do we, so let's take a look at it. We have TCT, AAC, TTT. Now we have to consistently refer back to this chart. Now let's look at the mRNA. Now this is where it becomes important. It's and why scrap paper becomes useful. I'm ignoring what came before it because they stayed the same. I'm assuming nothing's changing there. So over here, this would be AGA, UUG, and AAA. So AGA is arginine. So it was AGA was let me look at it, see what it was. Yeah, so it was arginine, so that's correct. Now, I changed it to serine. How do I change arginine, AGA, arginine? How do I change it to serine? Well, the easiest way is to get it to go AGU or AGA. C. Either way, AGU or AGC would give me a serine. Correct? Now, what if I instead, if I did that though, if I just substituted this position here, just substituted it, then that wouldn't get me the change after it, right? Because we said that after this, we'd get serine, right? If I change this position here, and then this RNA changed, I would get serine, that is correct. But after that, I would still get amino acids. And in the problem, mutant 3 doesn't have an amino acid after it. So this wouldn't solve the problem. But what could do it, if you think about it, what could get a serine is if we deleted this. What if we deleted this? If we deleted T, then this one becomes TCA, right? So this RNA becomes AGU, and AGU is serine. And then we have ACT. ACT then so now remember everything got shifted, right? So now this became this part of this one. Excuse me, you should be setting up for the test and being quiet. Please be quiet and set up for the test. Please be quiet and set up for the test. So that's TCA. Then this becomes the second one, right? So what's ACT turn into? That would be UGA. And what is UGA? It's a stop. Now that works, doesn't it?
Yeah.
Because then I would get this one turning into serine. This one would turn into serine, and this new DNA, because of the friendship, would say stop, so that I'd only get these 3. So that's definitely a deletion. Does that make sense? Look at how much work there is to this one problem. Right? Isn't that crazy? All right. So good job. I'm gonna post this today. And if you want to come back, if you have another study hall or lunch, we can go over the rest of this.
Okay.
All right. So here again, they give you the chart, the rectangular chart. They tell you the history of the chart. Beautiful. Then they tell you students investigate effects of a single base mutation. So one of this is the DNA. Let's write the DNA down. TAC AGC ACC TAG GCC. Is that correct? And then there's ... because it keeps going, right? And they say there's a single base mutation that occurs in a piece of the DNA. The diagram shows the first 15 bases of the DNA template they're investigating. A thymine nucleotide is mistakenly added between the A and the C. So what would happen is you'd have TAC AGC. Then you'd have AT CCTAGGCC ... Does that make sense? It just you added. So this is what's called an insertion. Correct? Because you're inserting a letter. Which mRNA sequence is produced when by the mutated DNA template? So you have this. This is you agree that this is the mutated one, right? The mutation is called a point mutation. It's an insertion mutation. Insertion mutation. But it's a mutation nonetheless. I am about to give you zeros for today's work if you're going to keep talking. Only thing you have to do is be quiet and work on your practice test. That's it. No questions right now. I'm in the middle of an explanation. I'm recording this whole discussion. which I would really rather not have on, but whatever, it's going to be on YouTube. So you have what's called an insertion, all right? So what's the new, it says, what's the new mRNA? Which mRNA sequence is produced by this mutated DNA? Well, that's kind of easy, isn't it? So I inserted it, right? I inserted my T. So now I'm going to get in my mRNA, I'm going to have T is going to be an A, correct? A is going to be a U. C is going to be a G. A is going to be a U. G is going to be a C. C a G. A is going to be a U. T is going to be an A. C is going to be a G. C is going to be a G. T is going to be an A. A is going to be a U. G is going to be a C. G is going to be a C. C a G, and C a G. So now that would be my new mRNA. Now I would divide it, I personally would divide it up because usually you want to do it by threes, right? Because it makes it easier to see it for me. There's going to be one extra letter. So which one of these would it be then? Don't say it, yeah, don't, just do it. Don't say it because I don't want people just to memorize things. Let me see. That looks right to me. Does it look right to you? All right. Why is it not the first one? How do you know that the first one's not the mRNA?
Because it doesn't go with T.
That's right. And there are no T's in RNA. So it can't be the first two. They gave you an easy way of figuring it out. If there's a T, it's not RNA. So if you read carefully, you know it's not the first two. That's a 50. It gives you 50-50 chance right away. Now move it up. It says, select all the effects the mutations will have on the protein produced. So what's going to happen on this protein? Well, we'd have to see the effects, we'd have to translate both the mRNAs. So this is the mutant, right? Just to be clear, to be absolutely perfectly correct, the best way to do it is to translate both of them and then transcribe and translate both the mutant and the normal, and then make the decision on what the changes were. Does that make sense? So the non-mutant or the normal, I'm just going to call it normal or average, TAC would be AUG, it would be UCG, it would be UGG, it would be AUC, and it would be CGG. So that's what it would be. Now let's translate. That was transcription, right? So now we have our mRNAs. I'm going to go ahead and do our little codon, my division, so I know where my codons are. All right. So AUG is methionine, because that's the start for both. So for the mutant, it's MET. You know what? Just to keep the color coding the same. I know it's a little bit OCD, but I'm going to go ahead and label it blue. So MET, UCG is serine, SER, correct? Do you know how to read that, yeah? Okay. UAG is stop. UAG stop. So stop.
Wait, you reread it like you go like this, first letter, second, and then you find a Greek letter.
Exactly. So U, A, G is what?
Stop.
Stop. So what does that mean? That's not an amino acid. That means what? Stop means
Stop.
stop.
Yeah.
No further. That's it. So this amino acid, this DNA sequence gives you an amino acid that's what? 2 amino acids long. A protein is 2 amino acids long. Does that make sense? All right, so let's do the next one. So in this one, you have AUG, which is methionine. Okay, that's fine. UCG, which is serine. UGG, which is tryptophan, TRP. What is it? I don't know, I'm confusing myself.
I think
So that was a mutant. That should have been
think
I'm
it
sorry.
meant to be a C.
Let me see. It's AU. Let's double check. Yeah, that's a C. So AUC, which is isoleucine, and then CGG, which would CGG, which is arginine. So we made the change was we made it longer, correct? So this insertion made it longer. So now let's go ahead and check what options we want to check off in these boxes. First one says the protein will be shorter. Is the protein the norm? I'm sorry. I did them backwards, didn't I? This was the normal, and the change happened in the mutant. What did the change do?
It made it stop.
It made it stop, so it made it shorter. So it did make it shorter. So click on that one, right? Do you agree? The protein will function differently.
Yeah.
Yes,
so
for
stop.
sure. Yeah, because it's shorter. The protein will gain additional active site. It's not going to gain anything.
Yeah, 'cause it stopped.
It stopped. All right, so that's deleted. Go ahead and delete it.
You can't delete it.
I mean, push the check. Oh, wait, I'm sorry, you're right. You're not going to choose it. Correct. The protein will have a more complex structure. No, it's simpler. The protein will be the same length, but will have different amino acid sequence. No. So you agree that those are your options? Now, see, look at how much work that took to do for this one problem. Now, be careful, because I did it backwards, because I didn't know what, I did the first part, and the first part only asked me, I didn't have to work with the normal, but in the second part, I had to work with the normal. So I almost confused myself, and I almost was looking at this as being the first, and then that, I almost looked at this being the first, and then this being the second. But this is the normal, and it changed into the mutant. So this is how it started, and this is how it ended. So it didn't get longer, it got shorter. Does that make sense? All right.
Okay.
Go
Thank
ahead.
you.
You're welcome. So it says a geneticist examines part of a gene in 4 different related species. So there's these 4 species, and they're related to one another. So you'd expect their DNA to be similar in some way or another. For each species, the bases in the sequence are indicated as follows. All right, so they give you a species list, they give you a little table, and one they have a species, and then another column is the DNA. I don't know if I'm gonna have to write it all out, but if I do, I will. And there's 1, 2, 3, 4. So there's 4 of them. And they said then she determines that the common ancestor of these 4 species has the strand so the one that they all share that was there. This is an unfair question that I added to this because we didn't discuss common ancestors and cladograms, and this is kind of what they're asking you to do.
Okay.
So if they have a common ancestor, that means that they have to share a lot of this DNA, right?
Right.
So it's like, if you're my cousin, right, then our grandfather, we probably share a lot of DNA with our grandfather,
Right.
both of us, all right? So that's kind of the idea. So A, C, C, T, A, G, G, T, G, A, G, C. Correct? That's our ancestor. So let's look at these these guys. Read me the sequence for 1.
A
Hold on, switch for me. A
C A G G T G T G C.
All right, give me the next one.
A C C T A G G T G T G C.
All right, next.
A C C A G G T G T C C.
And the last.
A C C A G T T G
TT what?
G T C C.
All right. So when we compare these, to me, well, first let's look at the question. Based on the data, which species is most closely related to the common ancestor? So you want to know which of these is most closely related to this common ancestor. What do you think you're going to do?
Match them up.
Match them up. And which one's the one that you're going to choose that's most closely related?
Yeah, that's why I picked the second one because it's like
It's the
same
closest.
until it gets to like the end.
It's the one that's the closest, right? So let's. I'm just going to verify your results, all right? So I read ACC as the first three. I see ACC here, I see ACC here, I see ACC here. Already there's one difference here, right? This top one has one difference.
That's the C. What A?
That A was different, right? The next series says TAG. It says TAG. Oopsie. TA. Oh, come on. TAG, right? So now I have AAG. TAG is the same. That's different. That's different. So the second one is matching at least those first two, and this guy is different here, and these three are different here, and then this TAG, it's AGG. This is a difference here. This is the first difference here, right? So there's only one difference so far, and this is only one difference as well. All right? Does that make sense?
Yes.
All right. So the next one says GTG, and the next one here is GTG. That's the same. GTG is the same here. GTG is the same here, and this is TTG. So this is actually This is a difference here. This is different.
Second difference.
That's a second difference. Correct. And then the last here is, I'll just go back to yellow, ACG or AGC. That's different, that's different, that's different, and that's really different. All right. So nobody shares AGC in that position. So the one that's the closest, you're absolutely right, is this one, right? Because it's the most closely related to this one. How? Because they share the most amount of DNA. The sequence is the same. Which one's the most different?
It would be the last one.
The last one. They both 1 and 4 have two differences, but the last one's also even longer.
Right.
And then on top of that, which of these 4 are most closely related?
It would be 2 and 3.
2 and 3 probably. So you can see how you can see that, right? All right, so that's all there is to it for this question. So
Thank you.
good choice.